This video is for the first part of chapter 13 on host defenses, looking at the adaptive arm of the immune system. So let's get over to those slides. All right, so this is part one. We're just basically going to look at all of the different components, the ingredients that make up our third line of defense, and we'll see sort of where this location occurs, where the location and the structure of these different lymphoid organs are, and talk about receptors. And then I'm going to kind of bleed this into the next sets of slides, which is looking at the function of the adaptive arm of the immune system and how T and B cells become activated. So bear with me as we go through this. I'm going to draw a whole bunch of diagrams, but of course we have a limitation with our technology here. So I'll be drawing on the whiteboard and sharing some images with you, and we'll see how well we get through this. Again, we'll start with this overview of the third line of defense and to talk about these different components of it and discuss the receptors on the surface of our B and T cells and our presenting cells. The major concepts for this entire chapter is basically the entire chapter. So really understanding development to response of both the B and T cell lymphocytes. So here we are, where we discussed already from chapter 12, we discussed the innate arm or non-specific arm of the immune system. We covered the first line of defense, talking about the physical and chemical barriers that protect us on a regular basis, our epithelium, our mucosa, our skin, of course, to protect us from any invading microbes or damage from the environment. We talked about the second line of defense, all the cellular components and also what they do. So talking about the process of inflammation, phagocytosis. And then we also mentioned sort of a lead in to how inflammation can trigger essentially or lead to the activation of the third line of defense, which is the acquired or specific response. Now, everything we've talked about so far has been the non-specific response, right? So everything essentially activates the innate immune system relatively the same with particular patterns. So viruses versus bacteria versus, gram-negative versus gram-positive or helminths, etc. And we talked about ways that you can evaluate that by looking at CBC counts, for example, and looking at the elevation of particular types of granulocytes or lymphatic cells, lymphocytes. So now we're going to discuss, though, the development of that highly specified, what I like to call the clonal army response. I always think of that Star Wars movie. And that really is what it is. So it's a development of a very fine-tuned, highly specific, well-trained army that is produced in mass quantities as a response to infectious agents and is usually highly effective at eliminating any kind of pathogenic infection. So for this one, we have a lot of things to talk about before we get to the actual response itself for the B and T cells. In order to understand how our B and T cells have our highly specified response, we have to discuss how they get there in the first place. So this is the broad overview, and I've just taken this essentially, there's a really great image from your chapter textbook, which is the next slide, and I've kind of just broken that down into each of these 4 different stages, and that's how we'll process through this whole thing. So first, lymphocytes are developed and gain their differentiation or their ability to respond to just about any kind of foreign antigen in the primary lymphoid organs, and that would be the bone marrow or the thymus for T cells. So we'll talk through this process and just really briefly touch on it. There's a whole field of science just studying lymphocyte development and differentiation. So if you're curious, I can lead you to more resources on that. But once lymphocytes gain their initial development, is essentially what we're talking about here is the development of B cell receptors or T cell receptors, immunoglobulins on the surface anchored to B cells or T cell receptors with their co-receptors too. And once they have this development of their receptors, and we call this your immune cell repertoire, so all the varying types of receptors that can be produced in order to provide us the ability to respond to just about any type of antigen or foreign molecule that comes into the body in the entire world, which is a crazy concept. This all happens before that pathogen comes into the body in the first place. Once that pathogen comes in, and this is really where we're focused on for our third line of defense, is when we have a pathogenic infection. Now we have our inflammatory response, all the stuff that we just talked about from that last chapter. The priming of our of our antigen presenting cells after they respond to that site of injury or inflammation. They pick up all that phagocytic debris, they process it, and they're taking it now to present the foreign particles, those antigens, to our lymphocytes in the bone marrow, or in the lymphoid organs like our lymph nodes or our spleen. So that's presentation of antigen. And this leads to, usually first, we're talking, at least in our discussion here, we'll talk about T cell challenge first, and the T cells lead to the challenge of B cell lymphocytes and then the development of the response. So here's a couple, the key understandings here is that there is a difference in the response based on T cell challenge versus B cell challenge. For B cells, the response always leads to antibody production. For T cells, we call this generally cell-mediated immunity. There's actually quite a number of various types of responses when T cells become activated, and it completely depends upon what the nature of that challenge is. So if it's, for example, intracellular versus extracellular pathogens, there's a different type of T cell response that will be produced, and we'll talk about that one in detail too. This is the picture from your book, the same thing, just in visual format. So again, these 4 different stages of development and activation. So the first step here is the development and differentiation of our B and T cells. Over here on this side, the B cell line starts as actually both of them start as hematopoietic stem cells, and then lymphocytic stem cells here in the bone marrow that then can further differentiate into B cells in the bone marrow, or can mature into T cells in the thymus. Both of these, once they have undergone differentiation and clonal deletion, which we'll talk about here, then we call them naive cells. They've never seen pathogen or antigen before, but they're primed and they're ready to go. They're released out of their primary lymphoid organ and will always migrate to a secondary lymphoid organ, either the lymph nodes or the spleen. And here they hang out until they come in contact with antigen. So here's that second part, that antigen-dependent response. Here's an example of a dendritic cell, which is key for activating our T cell, our first responders. This is what we talked about before, where there's inflammation. These dendritic cells are late responders, and they come and clean up that, that damage, essentially, picking up pathogens, digesting them, and taking those antigens to be presented in the secondary lymphoid organs like the lymph node. So here's a picture of this antigen presentation, and we'll draw this out in a lot of detail. This is showing you that T cells usually become activated first, and these can have a couple different responses. One is to help engage and activate these B cells, which then can turn into plasma cells, which is essentially the challenge, and then the response. These plasma cells then produce antibodies. Versus these T cells can also undergo different types of differentiation to become T helper cells or cytotoxic T cells, depending upon what the nature of that infection is, and become highly specialized, what we call effector cells, to go and do some specific function. So here's 3 different types of highly specialized effector cells: T helper cells, T regulatory cells, and T cytotoxic cells. So, and actually there's more, but we'll look at both of these arms of activation of our immune response. Some terms you should be aware of. So primary versus secondary. This is the first time you ever come in contact with the pathogen. That'll be your primary response. So this usually takes a few days, sometimes a few weeks, but that's kind of the exception. Usually it takes a few days for your primary response to become activated. This is your adaptive immune response, not your second line of defense, the inflammation. That stuff's always happening at all times, right? And you can have milliseconds for your inflammatory response to become engaged, but the activation of your adaptive immune arm takes a little while. However, after you have a primary response, remember we talked about this before, that you have a subset of your primary responding lymphocytes develop into memory cells. And these memory cells, this is your amnestic response or your memory response, where these things they stay in this not quite developed stage. So they're kind of primed. I know I used this word before, but they're partially differentiated. But the great thing about these memory cells is that they can become activated on a very, very short time frame. So the secondary response that you see here is essentially the second or any future time you come in contact with that exact same pathogen, or one that's very, very similar to it. Instead of taking days to activate your adaptive immune arm, now you have those memory cells that will become engaged right away and can respond. So that secondary response happens within usually minutes, maybe a couple hours, but usually within minutes, which prevents you essentially from getting infected with the same thing twice. Humoral immunity versus cell-mediated immunity. Humoral immunity is your B cell response. Let's see what color we're in right now. We're in red. Let's change it to white. Humoral immunity is your B cell response. Or antibody production, antibodies. Cell-mediated or cellular immunity, this is your, whoops, sorry, whatever. This is your T cell response. And there are various types of responses here. This can lead to cytotoxicity, so cell destruction, or it can lead to a variety of different types of responses. So we'll see that in more detail. And then these two terms, antigen, immunogen versus PAMP. Let's remind ourselves, PAMP, this is pathogen associated molecular pattern. And these are non-specific, if you recall. So any type of gram-negative lipopolysaccharide, for example, that would be a PAMP. And it doesn't matter whether it's from Escherichia or from Serratia or from Pseudomonas or from Enterobacter. It doesn't matter what the organism is. It's a shared associated, or it's a shared molecular pattern associated with gram-negative bacteria, and our innate immune arm has those pattern recognition receptors that can recognize general categories of molecular patterns. But now we're going to be discussing highly specific markers that can provide species specific or specificity. Specificity. Right, and so in this case, we can have one particular even subspecies, for example, of E. coli. E. coli is a really common organism, and there are strains that are non-pathogenic. They're part of our normal flora, excuse me. But there are strains that are certainly pathogenic and contain Shiga-like toxins, for example, like enterotoxigenic E. coli. And they have slight variations, right, to particular types of proteins. It's the same species, but there are subspecies differences. And these differences can be recognized by our immune system, our adaptive immune system, due to these variations to proteins or to components of the cell. And these would be considered these antigens, these little small pieces of proteins or other molecules in the cell that can give recognition on a species level. If they can stimulate the immune system, they're therefore antigens that are also immunogens, right? They can provide some level of immunity to you. So picture an antigen, again, any molecule that can bind specifically with any part of our immune system, our antibody or antigen receptor on a lymphocyte. If it can stimulate that response, it's considered an immunogen, and this is the idea here. Now, note that there's even more like fine picking of this thing. So this is a picture of antibodies that are binding to cell surface proteins. Now note that antibodies, they actually have a tiny little spot on the kind of arm portion, the FAB portion, that is able to bind to and recognize molecules. And that tiny part, it binds to each part on the antigen, on the protein. This is kind of like the children's song, like there's an ant on a log in the bottom, in the hole in the bottom of the sea. I don't know if you know that song, but it's just kind of like keep going down and down, finer detail until you see that tiny little thing. So an epitope is a region or a small part of a protein or a surface molecule that is actually where the recognition site for our antibodies or for our immune cell receptors is. So an epitope, we call it an antigenic determinant, right? So it's a really small region, maybe a few amino acids or a small region of a surface molecule that can provide the actual binding or docking station. It's kind of cool if you look at coronavirus research, and maybe in a minute I can pause the video and go find some images that I've been looking at, but they've not only identified the glycoprotein spikes that are on the virus that bind to our surface cell receptors, but they've identified on that the particular regions on the spikes that our immune system develops antibodies to that are highly efficient in neutralizing that virus's ability to bind to our ACE2 receptors. So it's not just about looking at, well, yeah, glycoprotein spikes, of course, it'd be great to, you know, reduce their ability to bind to host cells, so somehow send antibodies out to it. But antibodies are so tiny, you got to actually figure out which part of that giant glycoprotein would be the most effective at binding to neutralize that virus. So that's the epitope concept, the little part on that molecule. In fact, I'm going to go look for that just a second. Well, I found it for you. Let me go share that with you real quick so you can see what I'm talking about in real life. So this is a picture, and yeah, I have all kinds of things up there, but this is a paper that I was reading about the coronavirus, the current one that's plaguing our entire world right now. This is the spike protein, the actual glycoprotein that is responsible for binding to that ACE2 receptor. This is the receptor, but these images here are showing you this this docking station, or this part of the glycoprotein spike is what's responsible for binding to the receptor. And there are various, these are antibodies shown in magenta. So all of these different regions are identifying different epitopes on this antigen, this glycoprotein spike, that are great at neutralizing the ability to bind to that receptor. And this first picture here is actually how this glycoprotein spike interacts with our ACE2 receptors there. It's just this one small region here. So these different antibodies, you can maybe just imagine if this is the spot on the glycoprotein spike that is responsible for binding to our cell surface receptor, then the best antibody you could imagine would be maybe one that binds to the exact same region which this ADR antibody does and is highly effective at neutralizing or preventing that virus from actually binding to our surface receptors. These other antibodies have various levels of effectiveness, as you can see the epitope here. So even though the antigen is that glycoprotein spike, right, the glycoprotein itself, the epitope on that antigen can be different depending upon where that docking station is for the antibodies. So this is a great example of 4 different types of epitopes on the same antigen that actually confer various responses with the ability to neutralize this virus. Oh, actually, that's pretty cool. All right. Let's go back to our slides. Okay. Hopefully you're not getting tired of listening to me talk about coronavirus yet. Okay, so hopefully that concept is cemented in you that an antigen is really any kind of molecule. Oftentimes we're talking about a surface one, but it doesn't always have to be. An epitope, though, is a specific site on that antigen that is where we have the ability to bind to that with our antibodies or other receptors in our immune system. Whoops. Some examples of highly immunogenic antigens, so proteins or molecules that can stimulate a strong response. Lipopolysaccharide is probably the most notable one, but really anything here. So other glycoproteins that, like we just mentioned for viruses here, proteins, polypeptides like exotoxins, enzymes, anything cell surface is usually highly antigenic or immunogenic and can stimulate a pretty good response. It's a great question, and this is really more on thinking about like vaccine development. So I just want to give you a picture, and again, just making it super pertinent to what we're going through right now. There are a group of scientists, not a big one, but a group of scientists that are working on vaccine development very rapidly, trying to make the most effective vaccine that can help stimulate an immune response without the presence of the actual pathogen, right, inside the human. And I just gave you a clear picture of 4 different types of antibodies, 4 different antibodies for the exact same glycoprotein spike that actually show different levels of efficacy in their ability to prevent infection. And so and that's because there's different epitopes. So vaccine developers really have to figure out what the best epitope is on that antigen. So everyone knows, or most virologists are understanding, that the glycoprotein spike is really the best target for any vaccine development, trying to get your body to mount an immune response to that glycoprotein spike so that it can prevent the binding to our receptors and in effect halt that, infection or the pathology of it. But if you take that glycoprotein and just throw that protein itself inside a human can mount at varying levels of response to just that glycoprotein spike. So really trying to identify the epitope that's best and isolating out that epitope and giving that to a human essentially to provoke an immune response to just that one small region on the protein is where vaccine developers are kind of trying to focus. So if you're trying to identify this epitope, and you take an antigen and just denature it and provide a human, let's just say that they took that glycoprotein spike and just denatured it and threw it into a human, would you mount the same immune response to a denatured antigen as you would to the native or undenatured counterpart? And hopefully you can think about this. And even let me just go back to show you really quick, because I think this is a great example again. Just look at this, the incredibly complex folding pattern to this glycoprotein. It's a protein, right? And it's also glycosylated. So there's a whole bunch of sugar molecules all over this protein at different sites on the peptides, on the protein, different peptides on the protein. Specifically, asparaginins have a whole bunch of these different sugars on them. But look at the folding pattern here. If you were to take this protein essentially and unfold it and put it inside of a human, would you expect to see the same antibody response, even if you were to provide, I'm pointing at it, even if you were to provide ADR, for example, this region here, if you were to just take this region of peptides and just denature them and throw them in, hopefully you can see the answer is no. Epitopes are highly structure dependent, and that's just because of the nature of proteins, right? They require that secondary, tertiary, quaternary folding to form their final pattern. So if the epitope is here and you denature this, you're going to lose that structure. Even if you have inherently the same amino acid sequence, the epitope is determined usually by the structure and not the primary arrangement of those amino acids. So just to again, keep in mind what's actually happening. Okay. So this stuff is all occurring in our lymphoid system. We haven't talked about this quite yet. I know that this is actually from chapter 12, but I think it's more pertinent to talk about it here. So our lymphoid system is a really complex architecture or organization of tissues and different types of organs that their whole goal is to clean out any in any inflammation or damaged tissue as our immune system responds, and also bring together like a meetup site for our antigens and our lymphatic responders, our lymph nodes, our lymphocytes, sorry. So tissues and organs that bring antigens in contact with lymphocytes. You can kind of think about it as simple as that, but it's a lot more, there's a lot more to it than that. The lymphatic vessels themselves are the kind of what you would imagine would be like our blood vessels, right? The lymphatic vessels are similar to blood vessels, except of course, instead of carrying blood, they carry lymph. And lymph is this really complex concoction of whatever's out there in our tissue. So constantly things are draining from our tissue into our lymphatic vessels, and then they move through the lymphatic system they drain to these regions called lymph nodes or secondary lymphoid organs, where they can kind of be pass through a filter of T and B cells, and if there are antigens in there, those T and B cells may become activated at that point. It also allows our body to separate out any blood that has gotten through into our tissue and pull them out and put the blood cells back into the blood where it goes, keep the white blood cells perhaps back into the blood if they haven't been activated, and then removes proteins and things to help clear up our system. So the lymph itself is a bunch of our leukocytes that have arrived for that inflammatory response. Any antigens that were there are going to be in that lymph, and then any blood that has arrived. And if you recall from our conversation about inflammation, then you of course have edema built up at any site of inflammation as those blood vessels get leaky there. So you definitely have some infiltration of blood to those areas too that need to come out of the tissue, and they do so by traveling through the lymph. There is a one-way passage. Instead of blood going around and around, well, I guess as blood flows in one direction, I guess I could say, lymph also flows in one direction. Just as for our circulatory system, we have valves that open and close, and these are one-way valves to prevent any backflow. Same concept for our lymphatic system. There are valves that are one-way valves to continue the forward movement or moving things in one direction. Now, unlike the circulatory system that essentially starts at the heart and moves out with things collecting back up again, our lymphatic system, the one-way draining system, is essentially from our extremities, from outer regions where lymph gets inside lymphatic vessels, and all of it is directed towards one localized area. Not one central area like the heart, but a localized area depending upon the proximity or depending upon where the draining is. And let me just show you a picture, it's much easier. Okay, so here's a picture of the circulatory system. We know that there's a central region here and everything is pumped out and back to the heart again. But the lymphatic system is a little bit different. The direction is essentially from the extremities here, and all of these things drain to one localized region, whatever secondary lymphoid organ is closest to that draining region. So for example, when you get an upper respiratory tract infection, when you get a cold, when you develop coronavirus, any of those things, you'll feel, and we all know this, your throat gets really swollen, not just from the inside, but on the outside here and behind your ears. This is my son's most sensitive spot in the world, is behind his ears here. And the reason that you get so swollen here is because you're experiencing an infection up in this area. And so your lymph vessels, all of your lymphatic vessels up here in the upper respiratory region in your sinuses, they all drain to these central, what are called cervical nodes here. Your tonsils are them too, but your cervical nodes. And these are essentially where all of that inflammation is happening, all of that pus formation. Whew, I'm gonna sneeze again. All of that stuff drains right there into your lymph nodes, and so they get swollen as not only you have an influx of fluids draining from the lymphatic vessels, but also this is likely where your B and T cells will become activated and respond and proliferate, right? So they actually develop and expand and form these large expansive centers called germinal centers inside your lymph nodes, and so they'll start to swell both from the influx of fluid but also from the development of an appropriate lymphocyte response. Gosh, my vocabulary today. So it's kind of cool. I mean, so we have these specialized regions. There are some in your throat here. There's some down underneath your armpits and in your groin. Those are the primary regions. And then of course in the central cavity here, we have abdominal nodes. And then we have our spleen, which is another very key factor in systemic immunity. So when you have something come into your blood, then usually the spleen is the first thing to respond right away. And it does not usually start here in these outer secondary lymphoid organs. So it's a really extensive system, a pretty amazing system, but just understand, so all of these lymph nodes are considered secondary lymphoid organs. They're a collection of the influx of lymph from draining lymphatic vessels. And then also there's circulation that goes right there too. So there is a connection between our circulatory system and our lymphatic system. And that'll become really important as we talk about the development, like where our T and B cells come from and where they go, and then how they become activated. So our lymphocytes do need to travel through the blood. So it's important that there is a connection between our circulatory system and our lymphatic system. You'll hear someone singing in the background for a little bit. Okay, so again, some terms for you. Just a second. Okay, some terms for you. Our primary lymphoid organs are where we have the development and differentiation of our lymphocytes, so the maturing of our lymphocytes. to prepare them for the presentation of antigen or the ability to respond to antigen. So this would be either the bone marrow for all of our lymphocytes, B and T cells, but our B cells stay in the bone marrow for the final development of their B cell receptors. The T cells migrate to the thymus, which is where they undergo their thymocyte boot camp essentially and get their development of their receptors. So the primary lymphoid organs are the bone marrow and the thymus. Secondary lymphoid organs are where our T and B cell lymphocytes go before they see antigen. So they're naive when they leave their primary lymphoid organ, and they go and they hang out in these secondary lymphoid organs, and they wait essentially to become activated. If your body needs them, if they have the particular types of receptors that recognize that specific pathogen that's coming into the body, then they will become activated in the lymph nodes, and then most of the time they will leave and become effector cells, is what we call them, and head out to the actual tissue that's being infected. If they don't become activated, usually these guys only last a couple of weeks. And so there's a constant turnover of our B and T cells. If you can imagine, we there's an indefinite number of, not quite, but there's seemingly an indefinite number of antigens that can come into our body on these different pathogens. So if we had a limited number of B and T cells that we produce and they just stayed there forever, perhaps we would not have the army, the first, the repertoire is what we call it, but we wouldn't have the ability to see whatever that is that's coming inside the body. So there's constant turnover where new B and T cells are constantly being made that have variations to their receptors, just in case, well, so that we have the ability to detect anything that comes into our body. So if they don't get activated, they die, but if they do, there's a very specific pattern behind this activation. These are meetup spots because not only do the lymphocytes hang out here, but also this is where incoming lymph from infected tissue comes and drains here, and with it comes antigen that's kind of free floating in that lymph, but also importantly, antigen presenting cells. These macrophages are dendritic cells that have been primed. They've eaten up these pathogens at the site of injury, and they're presenting antigen on the surface of their cells. And so they're going to come in and they have a directed path through that lymph node or that secondary lymphoid organ in order to activate those lymphocytes. Examples of these, lymph nodes are what we're going to talk about the most. The spleen is actually one of the most hyperactive ones. So this is anything that comes into the blood will activate the spleen, the lymphocytes that are in the spleen. Peyer's patches, this is what's found in the gut, and tonsils are in the throat. Now that's just a tiny, tiny representation of these gathering places, these secondary lymphoid organs. In fact, every single part of the body has kind of a specialized secondary lymphoid organ. The respiratory tract, the throat, even up here, your colon versus your small intestine. So we have mucosal associated lymphoid tissue and skin associated lymphoid tissue, which is what you see here, malt and salt. Galt is actually our biggest one, which is our gut associated lymphoid tissue, right? All of this is lymphoid tissue. So these kind of meetup spots for our second and third lines of defense. Now I'm going to show you a picture of a lymph node because that's how I'm going to focus on the second sets of slides. We're just going to write out a lymph node and show the process of T and B cell activation at the lymph node. This is a picture of one here actually from your textbook. This is a cross section, an electron micrograph of a lymph node, and it highlights some of these key areas here. There is very specific orchestration of these lymph nodes. You can kind of see in this electron micrograph, these invaginations here. These are the connections essentially between the entering lymph and the medulla oblongata, medulla, and essentially where these different zones are. But note here, this lymph node is divided into highly specialized regions. So on the outside, we have these afferent vessels, that's where the lymph is coming in. The lymph comes in, and remember, it has white blood cells, it has some red blood cells, it has antigens. Of course, those white blood cells are in the form of antigen presenting cells primarily. So they've got to get to their T cells in order to activate them first. And so there's a directed path essentially of lymph that kind of percolates through this area and gets to these T cell rich zones down here. There are other regions here that are on the outside. This is the cortex. These are mostly where B cells are hanging out. These are called follicles, and at the center of follicles are the germinal centers where you have actively replicating B cells that are getting ready to differentiate into effector cells. So you have these highly specialized regions, and their architecture is important in understanding the organization. So I've added an extra image here just so I can start the conversation about how this lymph node works. And recall, this is just one example of organized lymphoid tissue, but this is a really, really great example of how our third line of defense becomes engaged and begins the process of activation. So again, we have these these very specialized regions. The lymph enters from tissue in all these kind of external regions. These are called afferent vessels, and the lymph will leave through efferent vessels, right? So afferent and efferent. The outside ones that bring lymph in are the afferent vessels. They bring in all the stuff. So here's an example here of some of the stuff that's coming in, including our dendritic cells from our tissue, at least out there, right? So this lymph is coming in, and then it drains in a very specific pattern. So here's a picture of, in yellow, the lymph flow. An afferent lymphatic vessel brings lymph in, and it sort of migrates through what we call the sinuses. And I like to consider this like a percolator. If you guys don't know what that is, it's an old-fashioned coffee pot that some people still use, where essentially water can come in at the top and percolate through, or just kind of moves through all of the grains of the coffee to give you coffee at the end. So that's what happens to this lymph flow. It comes in through the afferent vessels, and then it moves through these sinuses and starts to percolate through. So it kind of migrates down into the medulla, and then it just moves through these complex vessels there until it's kind of filtered out, and then it's collected here in the efferent vessel and then kind of spit back out into the body. During this process, antigen is coming in, as well as antigen presenting cells. Now note also we have blood vessels coming into and out of the lymph nodes, and this is important in bringing in our what we call naive or primed T and B cells to get to their regions that they hang out in. So this is how our lymphocytes get here. They come in through something called high endothelial venules, and they're stuck here either at the germinal center for B cell, or at the follicles for B cells, or at the T cell zones for T cells. So you can see in this image right here, in this little node here, the different locations for these B and T cells. So B cells really do congregate in one region, these follicles that are in each of these different regions. And then the T cells move down here into the T cell zone. This is kind of, there are specific vocabulary words for each of these different parts of the lymph node, the anatomy of the lymph node, but you don't have to know about all of these different terms here. You do need to know that B cells do migrate to the follicles here, and the T cells kind of hang out in the T cell zone, and that lymph kind of moves in this one-way direction, percolating through the T cell zone here, which allows activation of the T cells, and these guys can come and communicate with the B cells at the follicles. So let's start at the top here with lymphocyte development and differentiation. Okay, lymphocytes all start as pluripotent stem cells, as you recall from our last chapter. These are all in the bone marrow, and these pluripotent stem cells can differentiate into red blood cells or white blood cells or platelets. Of the white blood cells, we have those granulocytes that we discussed in our last chapter, neutrophils, eosinophils, basophils, etc. Monocytes, which can then develop into our antigen-presenting dendritic cells or macrophages, and our lymphocytes, our B and T cells. And we're focused, of course, on the lymphocytes. These then further, the lymphocytic precursors or lymphocytic stem cells can then further differentiate into either T cells or B cells. And then we have the T cells move into the thymus to gain their antigen receptors. B cells will stay in the bone marrow to gain their receptors. And these guys, once they have built their receptors, they move out of their primary lymphoid organ and end up in the secondary lymphoid tissue. Here, like the lymph node that I just showed you, some will self-destruct, but others will become activated, and usually they will leave depending upon what the response is. Some will actually stay and do their effector function in the lymph node, but we'll talk about that more in the next set of slides. But most will leave and they'll go and do their function out in the tissue where there is that infectious agent. Again, just to reiterate, all of them come from that exact same pluripotent stem cell, and then lymphocytic stem cell for B and T cells. B cells will mature in the bone marrow, T cells mature in the thymus. So we're going to take a little bit of time and talk through these receptors and start with these general all-purpose receptors, but understand what the purpose of these things are. So receptors confer specificity and identity. They can give your body the ability to recognize self from non-self. There are many different functions of these things. We've already talked about how important receptors are in that cell-to-cell communication with cytokines or chemokines. There we go. So there's one of them, receiving and transmitting chemical messages. Of course, attaching to foreign antigens, that would be important for immune cells. For our epithelial cells, we do have those types of pattern recognition receptors to do the same thing. But our immune cells are highly adapted to do this. So we have many different types of very key receptors that are able to recognize and attach to foreign material. We do on many of our cells, specifically our T cells. Our T cells have the ability to recognize self cells from non-self cells, and again, becomes important to know whether to destroy that cell or to not. And then in cellular development, we talked about this in our last chapter where we were looking at that pluripotent stem cell, and I mentioned that that process of differentiation where that stem cell turns into, you know, one fate or another fate, a white blood cell or a red blood cell, for example, that differentiation comes from key chemical messages that are sent by the body. That would be hormones or cytokines from even your immune cells or your damaged epithelium. And those things then will aid in that cellular development to really trigger or push that stem cell in one direction or another. This is a picture of both T and B cells and looking at many of their surface receptors. Don't get daunted by this. There's actually not too much going on here, although again, this is a very simplified picture. So let's first look at the T cell surface. T cells have these very specific types of receptors called T cell receptors, but this is a receptor complex here. So T cell receptors have one receptor that, or one region that is really important in binding to the actual pathogen, the antigen binding site. But next to this, there's also these co-receptors called CD3 co-receptors, and then either a CD4 or a CD8 co-receptor. Now the CD3s are important in anchoring this receptor complex and also in helping with cell signaling, versus this CD4 co-receptor. We'll talk about that in a lot more detail in just a second, but this is important in helping recognize what type of cell is actually presenting that antigen. For B cells have both our non-specific MHC markers, which we'll talk about next, and specific antigen receptors here in the form of an immunoglobulin, an anchored immunoglobulin, and usually IgD. These things have that antigen binding site here, which provides them the level of specificity that we see here. In fact, even more diverse repertoire than T cells can attain. And then they have the ability to present antigen also, and we'll see that in our next set of slides. Let's start here with the MHC complex and explain the concept of antigen presentation and self from non-self. So the MHC complex, this is a large set of genes that encode for a single glycoprotein receptor. And in fact, we call this a human leukocyte antigen because every person, their cells at birth, or I guess, of course, before birth, in utero, our cells, maybe even as early as that fertilized zygote stage, will essentially put together a piece of their own glycoprotein receptor from a variety of these different genes to give us our own unique signature of this particular receptor. So everyone's MHC receptor, their MHC molecule, is, although we all have the same basic set of genes, the particular combinations of those gene segments for each person actually is different. And so once your cells, upon epigenetic determination way back early on, determine that set of genes that's going to be your MHC complex. Every single cell that is made in your body, except with the exception of red blood cells, has an MHC complex on it, MHC molecule. So we call it again that human leukocyte antigen, because every person inherits a unique combination of these genes to make this class 1 antigen. So there are 3 classes of these MHC, or major histocompatibility complex molecules. Let me pause for a second, actually, because what I want to do also is help you with, oh, I haven't even done it yet. Okay, let me pause this for just a second. I'm going to pull up our abbreviations chart that we were working on before and add to that. You guys have okay. Okay, so I just wanted to share that I know that we were working on this abbreviations chart. We have my exam up there too. Let's work on continuing our abbreviations. So we'll add a few that we had some before, and we'll add a few here. Just as a reminder, we'll talk about macrophages like this. It should be a phage sign, but we'll do it like this. Macrophage. APC, we already know, is an antigen presenting cell. We've learned some new terms here already, actually. AG is what we're going to use to describe any kind of antigen. And when we're talking about lymph nodes, usually we say lymph node. I think that we're ready now to move into what we're talking about here. So when we're discussing receptors, MHC receptor is called, this is the major histocompatibility complex, right? And we have both MHC class 1, which determine, self, and MHC class 2, which are APCs. Okay. All right, so MHC class 1 is responsible for presenting endogenous or self antigens. Let me explain that for just a second. Let me go to our whiteboard. All right. Okay, so let's say that this is one of your own cells. Actually, a really great way to think about this is, let's say this is a macrophage or a dendritic cell. Now, at any given time, of course, we have our nucleus here with DNA. Now, we are always making transcripts, and those transcripts are getting transcribed into proteins, right? Protein. Now, inside our cells, we have this recycling machinery that is responsible for taking a small little subset of our proteins and breaking them down. So we have unique machinery that takes these guys and digests them, and for the purpose of presenting them on the surface of our cells. And the loading of these things usually is done, let's say we have of course, our endoplasmic reticulum here, and we have our Golgi apparatus way out here. And this is our rough endoplasmic reticulum. We have proteins are made, most of them are made inside our rough endoplasmic reticulum, right? And this digestion machinery, I think this is in our cytoplasm, but there's a very fine-tuned way that this digestion works. Now, inside these guys, there is the development of these particular receptors, our major histocompatibility receptors. And they're usually made here at the rough endoplasmic reticulum, and they're sent out in these vessels that then go and fuse with our membrane, and we have the presentation of our MHC receptors. Let's do these ones in green. So there'll be these receptors that are made, they're kind of embedded in this, thing. And then when this endosome fuses, then we have now our MHC receptors that are all fused to our membrane. Now these MHC receptors, their job is to, I should have put these over there, but their job is to essentially take these digested protein pieces and let me erase this part right here. So their job is to take these protein pieces and load them onto these MHC receptors. Okay, so these digested protein fragments are then loaded onto each of these guys here and presented on the surface. I wish that I had done all of that stuff not in red. Let's do it all in blue instead. I'm just going to change the color here. I forgot that I usually do antigen stuff in red. Okay, here we go. So now we have blue. These are self antigens. Self. This is an important distinction. So these, our proteins are then broken down and little self peptides, little self pieces are loaded onto these MHC complexes, and then now they're on the surface. So in blue is considered a self antigen. So a piece of something of our own, our proteins that are made or other components that are digested by internal machinery for the purpose of generating these little fragments that are loaded, these peptide fragments that are loaded on MHC class 1 complexes and presented on the surface. So what I've just drawn here are MHC class 1. And the reason that this is done is to continuously alert the body that this cell is only producing self proteins. Now, why is that important? Let's say, now I'll do my red stuff. Let's say we have a virus, right? Like coronavirus or something, which of course I'm drawing naked, not an enveloped virus, but whatever. Let's say we have a virus and the virus infects the cell. Remember that the job of a virus, once it gets inside, right, it uncoats and it causes the host machinery to now make its own proteins. So there's a whole bunch of viral nucleic acid and viral protein parts that are being made now by the host cell. Now the host cell will actually take all of these proteins, since it's making it, and will do the same exact thing. Not only will it do the job of the virus by making more virus parts, but it also will take the viral protein pieces and do the same thing it normally does. Digest them, throws them onto MHC class 1, and then presents them. This time though, of course, the MHC class 1 molecules, instead of presenting self, or in addition to presenting self, they're now going to be presenting viral pieces, right? Viral antigens. So this is considered a foreign antigen and still on MHC class 1. So let's say, again, this is a picture of a viral infected cell. And so when a virus infects or an intracellular pathogen infects the cell and starts to produce their own proteins or has the host produce proteins for them, those proteins are still going to be digested by that internal cellular machinery for this purpose of surveillance. But now, instead of having just self-antigens on the surface of the cell, this infected cell is now essentially flagging down the immune system by saying, hey, I'm infected. There's this foreign stuff that I'm now presenting out here. And our immune surveillance network, their job is to constantly survey the tissue. We talked about that in the last sets of slides, that the purpose of the immune system is surveillance. That's one of the biggest ones. Under steady state conditions even, is to survey. That means they're constantly going through tissue, they're going through blood, they're looking for any signs of infection or something foreign. And this is a way for our cells to say, I am no longer healthy, right? This is really important for viral infected cells. Also, you can imagine maybe cancer cells will do this too, where they begin producing some kind of mutated protein that doesn't look like what you're supposed to be producing. And that mutated protein will also still go through the same internal machinery, will now be presented on the surface of the cell in MHC class 1, and your surveillance system will come through and be like, I don't recognize that protein. That doesn't look like anything that I've seen before. And so this response, the presentation of foreign stuff on MHC class 1 is just an inherent way for all of our cells to be able to trigger or flag the immune system to determine or to say that something is wrong, right? Something is wrong. So under normal circumstances, your MHC class 1 receptors are constantly producing self-antigens or presenting self-antigens on those receptors. This is what neutralizes our immune system, says don't attack. I'm good, I'm healthy, stay away, right? When that is changed because of mutations or because of infection, and there now is foreign antigen presented on the surface, this is a call to alarm, right? And our particularly cytotoxic T cells, but we'll talk about that in just a minute. One particular arm of our immune system, their job is, their whole MO is to go and say, what is on your MHC class 1? Is it self? Is it foreign? If it's foreign, you have to self-destruct. If it's self, you can keep on living, right? So that's this concept of MHC class 1. Now, MHC class 1 receptors are on the surface of every single one of our cells, right, except for red blood cells. So all of our cells have this human leukocyte antigen. This is also what can lead to graft rejection. So because all of our cells, every organ, tissue, cell, everything, if you receive an organ from someone who's even very similarly related, a brother or a sister, and it goes into your body, everyone except your identical twin, every single person, even your closest brother or sister, has a different MHC class 1 receptor on the surface of those cells. So your body will then no longer be able to respond appropriately and will consider that cell as foreign if it doesn't have your own human leukocyte antigen on it. This is also why gene therapy, or the concept of taking out your own bone marrow cells, modifying them and putting them back in, is a very good idea, because you're only modifying what's wrong with them, but you still have your own human leukocyte antigen, or MHC class 1, that you're putting back into your own body, so you don't have to worry about that concept of graft rejection for whatever that modification is that you're doing. Okay, that's MHC class 1. MHC class 2 is another set of surface receptors responsible for presenting antigen, but in this case, it's always presenting foreign antigen, and these are on our antigen presenting cells. So this is MHC class 2. Okay, so this was MHC class 1 that we just looked at here. Let me, maybe it'd be easier if I gave you some quieter noises, more directed noises. Let me change and put some earbuds in. Hello, hello, okay, still works. Okay, MHC class 1. And this is an example of MHC class 2 over here. This is primarily found on only our antigen presenting cells, MHC class 2. So let me draw kind of an example of that with this dendritic cell. Dendritic cell, antigen presenting cell, right? And this guy also has these surface receptors, but their job, so during that process of phagocytosis, right, where we have that internal phagolysosome, we'll also have, and here's the nucleus over here, right, producing their own major histocompatibility complex. Let's do these ones in this teal. Oops, teal. Here is MHC class 2. And these guys will go and pick up antigens, foreign pieces that have been produced during this phagolysosome digestion, right? And they'll pick these guys up and form these complexes of MHC class 2 and foreign antigen. And fuse them here, so you end up with them on the surface of the cell. So now we have our foreign antigen being presented by MHC class 2. And this is for the purpose of antigen presentation. This will be presented to T cells in the secondary lymphoid organ and can lead to a very specific type of activation. So both of these ones are examples of very similar things, that there's foreign antigen being presented by an MHC complex, but they mean totally different things. Because this is MHC class 2, these are found only on antigen presenting cells. So this is a very specific signal that triggers the activation of a particular type of T cell, versus this is MHC class 1 out in the tissue, for example, with an infected cell presenting foreign antigen, and this triggers self-destruction by another type of T cells. So these mean totally different things. If antigens are foreign antigens are on MHC class 2, this is just indicative of an antigen presenting cell presenting antigen, and you'll find this happening in the secondary lymphoid organs. This, when you see MHC class 1 with a foreign antigen, with very little exception, and I'll explain that in a little bit, with very little exception, this, if it's out in the tissue, will always mean this is some kind of mutated or infected cell that is needing to be triggered to self-destruct, right? Because normally MHC class 1 presents self-antigen, and only when that cell is infected or damaged will it be presenting something foreign. Okay, so that's the concept of MHC classes 1 and 2. Now there is a third class which has to do with the complement system, but we're not going to discuss that one here. But just to be aware that this MHC class 3 is involved in activating that complement system. Let's see if this continues. This is a better picture. I know I'm just drawing them as really simple kind of 2 lines here, but there is a difference in structure for MHC class 1 versus MHC class 2. Your book has them demonstrated like this, where there's an antigen binding site or an antigen binding region right in the center of MHC class 2, versus MHC class 1 has an antigen binding cleft that's responsible for attaching to peptides inside that cell and putting them on. Again, MHC class 1 is on every one of your cells except for your red blood cells, so all of your nucleated cells, versus MHC class 2 is found on only antigen presenting cells, okay? Note also, because this will become important, I don't want to confuse you quite yet, but antigen presenting cells that have MHC class 2 also have MHC class 1. And this will become really important when we discuss the activation of our B and T cells, well, specifically our T cells. Okay, let's jump to B and T cell receptors. In fact, I think what I usually like to do is start with T cell receptors and then go backwards here, because I think it makes a lot more sense. So let's do that. Oops. I'm going to skip ahead to slide number 29. Okay, so let's look at the development of T cells and what they look like first, because we just finished talking about MHC receptors. So hopefully this will make sense as we walk through it. So T cells have a very specific type of receptor. There's two arms to this receptor. Both of them are exact copies of each other. And for each of these receptors, there's a variable region that's responsible for binding to a foreign antigen, and a constant region, which is where that membrane anchoring is. Now, I will say that although this is the simplified version of the T cell receptor, note that there's a T cell receptor complex that is a little bit more complicated. So let's look at this. So here again is a picture of that T cell receptor. There's a foreign antigen right here, some type of a small part of a protein, like a peptide, and it's being presented, in this case, it's being presented by an antigen presenting cell, and therefore this is likely MHC class II complex, binding to that foreign antigen and presenting it. So here's this beautiful match between the antigen presenting cell and the T cell, with its T cell receptor able to bind to and recognize with perfect what's the S word? Specificity to that foreign antigen. Now there's a whole bunch of other stuff that you see here, a couple of things that you do need to know. So here's that CD3 complex that's important in anchoring this and actually has the signaling molecules on it that's important in actually initiating a signal cascade for this T cell receptor. But then also over here on the side is this really unusual and cool receptor called the co-receptor. And this would be either a CD8 co-receptor or a CD4 co-receptor. Now this will tell you which type of cell each of these things is. So these co-receptors, again, CD4 versus CD8, this will tell you which type of T cell each of these things are. So let's go back to our whiteboard and we'll try to draw this out. Okay, so here we have our pictures of an MHC class II antigen presenting cell presenting foreign antigen on MHC class II. And here's a picture of an infected cell that has MHC class I molecules and that foreign antigen. So I'm going to draw just right on top of this what types of T cells would become engaged in each of these circumstances. So let's draw our T helper cells. We'll start with those. So we'll draw those in a lovely light green. So over here on the side, I'll draw a T helper cell. I forgot I have a stylus in my hand. Our T helper cells. These T helper cells these guys have something called a CD4 co-receptor. So we'll call them CD4 positive because they have that receptor. And then to abbreviate this, we'll call it TH. TH. That's a CD4 T helper cell. Let me add these terms to our growing list here, and I think I need to add a couple things. Okay. Okay. TH is T helper cell, and they have a CD4 co-receptor. And then these guys, the next ones I'll write are TCs, and this means cytotoxic, T cytotoxic cell, and they have a CD8 co-receptor. Oh, I know what I was going to add. I was going to add up here a dendritic cell. Okay. Let's go back. Okay, here we are with our T helper cell. This has a few important ingredients as mentioned already. So let's add in our T cell receptor. I should have drawn a little bit closer, but let's say that it's going to engage with this antigen presenting cell. And so here's our T cell receptor, right? T cell receptor. And this is the variable region, which is antigen binding. So if this is a perfect match where the peptide fragment that's being presented by that MHC class II is a perfect match here with that variable region, this will cause receptor engagement. But this T cell receptor doesn't really have a great way to do that. So the CD3 co-receptors here actually have signaling molecules on them. So these are CD3 receptors, and these are involved in signaling. But the part that I want to really emphasize is our co-receptor. I'll just keep it in the same color as we've been doing. So our co-receptor here for the CD4 positive T cells is the CD4 co-receptor. CD4. CD stands for, what does it stand for? Something of differentiation, cluster of differentiation. So this is it's a gene segment that produces these particular types of receptors. Oops. So the CD4 co-receptor is responsible for binding to and recognizing MHC class 2. Okay, so this is the complex that we're forming here. Let me highlight this here in pink. Okay, so what we just saw is, and let's try to keep it color coded. So on our APC, so we have our T cell here, and we have our APC here. Okay, on our APCs have antigen presenting cells have MHC class 2. MHC class 2s are recognized by, on the T helper cell, that CD4 co-receptor, right? They bind to and recognize MHC class 2. Now, MHC class 2s are presenting foreign antigen on that antigen presenting cell, and that foreign antigen is recognized by the T cell receptor. Okay, so this is the complex that we're seeing here. This is the complex that can be formed with these T helper cells in the lymph node when they're coming in contact with an antigen presenting cell. That antigen is presented on MHC class 2, and remember that this is from some site of injury or inflammation where there's some foreign pathogen that has been digested by this phagocytic cell. This guy has migrated to the lymphoid organ and is now presenting those antigens on MHC class 2. That presenting cell, that APC, will essentially percolate through the lymph node until it comes in contact with a CD4 positive T helper cell that has a T cell receptor that binds with perfect affinity or perfect match to that antigen. And if it is a CD4 positive T cell, it'll have that CD4 co-receptor that will bind to and match with that MHC class 2. This match right here, the CD4 co-receptor, actually tells this T helper cell that what it is binding to is an antigen presenting cell, and therefore it becomes engaged. So if this CD4 positive CD4 co-receptor does not bind, then it will not lead to engagement of the CD4 positive T helper cell, because both of these receptors, the T cell receptor and the CD4 co-receptor, both of them need to make this bond to what their ligand is in order for activation of the CD4 positive T cell. Because it's important that T helper cells are not activated by an infected cell, right? This infected cell, it needs to be destroyed. T helper cells, they don't do that. But another type of cell does, and that would be the cytotoxic T cell. Let's draw those in purple, sure, why not? So over here, we have a cytotoxic T cell. These guys have a CD8 co-receptor, and they're abbreviated TC, or cytotoxic T cell. Now these guys also have the same, sorry, let me try to move this thing out of the way again. All right, these guys also have the same T cell receptor that I drew over there. So here, again, is that T cell receptor with that variable region, so TCR, T cell receptor, with that variable region. These guys also still have CD3. In fact, CD3 is a marker of T cells. So if you were trying to just grab a whole pool of white blood cells and just pull out any possible T cells, you would look for CD3 co-receptor, right? Here's that CD3 signaling co-receptor. And now these guys have a cytotoxic a co-receptor that's CD8. And this one specifically binds to and recognizes MHC class 1, right? So here's CD8 co-receptor. Let's draw this little match made in heaven thing. So let's draw out this connection over here, just as we did before. So here's our cytotoxic, whoops. Here's our cytotoxic T cell, and here is our not APC. Well, no, I won't say APC because that's too confusing here. Oops. And this is our presenting cell. Let's say this is an infected cell. Infected cell. Because that's the picture that we're showing here, right? Let's say that we have a virus infected cell, and this viral infected cell is somewhere out in the tissue, and it is presenting that viral protein or peptides on the surface of MHC class 2. So now let's look at this receptor engagement here for these cytotoxic T cells. And let me just, no, I won't say it yet. Okay, let me go through this first, and then I'll say it next. Okay, in this case, we have our, just as we did before, we have our co-receptor that recognizes its ligand, which is MHC class 1, or self, right? These are our self receptors that are normally presenting self. Let me just draw that. No, that was a really bad one. Looks like 2. So MHC class 1. All right. At that receptor complex here, right? So we have that CD8 co-receptor that matches with MHC class 1. Here is the T cell receptor that, same as before, matches with that foreign antigen. So T cell receptor here that must have a perfect match with a foreign antigen. Now, it will not be self-antigen. Self-antigen does not cause this reaction because we have development to delete out any self-reactive. So this only will recognize foreign antigen. And again, when this complex is formed, when you have that CD8 co-receptor engaged with MHC class 1, and you have that T cell receptor engaged perfectly with some foreign antigen, this will then lead to activation. And also this will lead to activation. Okay, now since I'm drawing these pictures here, I do need to clarify one thing because I'll use these terms very shortly. This here is a picture, I'm gonna kind of draw a little line between these two things because I'm gonna say this is a picture of what's happening in the lymph node to initially activate your T helper cells. And these ones would therefore be considered naive, which means they've come from their primary lymphoid organ, the thymus, and they've entered into that lymph node and they're just hanging out and waiting for that first time around activation. However, what I'm drawing here, because this is an infected cell, this is in injured tissue. And if it's an injured tissue, then the type of T cell that I'm talking about here, or the state of that T cell, this guy is not in the naive form. And I'll show you how it gets activated in the lymph node in just a little bit, or maybe in the second set of slides. But for our purpose here, we have to say that this is actually what we call an effector cell. That means that it has already been primed in the lymph node by a similar mechanism as this, and it has left the lymph node and gone in search of the same little antigen that got it started in the first place. So it's going out in specifically as an army of highly specialized killer cells, and their job is to go and find where that infection is and just go and eliminate any cell that is infected with that same pathogen. Right, so a naive means it hasn't seen antigen before. This is actually the first time around, and this happens in the lymph node. Effector means this is after it's already been primed in the secondary lymphoid organ, and it is out in the tissue looking for that infected tissue. This is an effector. It's going to go do something, versus this is just getting primed in the lymph node. Don't be too confused on this yet, but I really want to just clearly emphasize that although I'm drawing these and everything is correct here, that the responses that you see are based on whether this is in the lymph node or the effector cell in the tissue. All right, so back over here. We just talked through these co-receptors, CD4 and CD8, and they differentiate which type of cell is doing the binding, doing the presenting, right? If it's a self-infected cell or if it is an antigen-presenting cell. This is a picture of a T cell, and again, there's that T cell receptor that has that transmembrane domain. There's that CD3 co-receptor and CD4, which is responsible in this case, if you recall, for binding to MHC class 2 on antigen-presenting cells. That means that this guy is a T helper cell. You know that because of its co-receptor, because of its ability to engage in antigen-presenting cells. This was CD8, we would call it a cytotoxic T cell, and we would know that this is going to bind to self-infected cells. Okay, let's actually reverse ourselves. We're going to go back and talk about B cells here. So I'm going to stop these slides for just a second. All right. Oops. I just moved it and then it didn't move. Okay, here we go. So B cell receptors are similar, but they have some unique differences. Let me draw this first. I'm going to switch over to that whiteboard, and hopefully you've captured that with a screenshot, or you can go back and pause the video and draw everything that I just wrote down, because what I'm going to do is change this. Okay. What I'm going to do is clear all this stuff here and write out some information about B cells, okay? Okay, there we go. Clear all my drawings. Here we go. Okay, B cells have on their surface a couple of different things. So they have B cell receptors, which are in the form of an immunoglobulin. Immunoglobulins, oh, just got chilly. Look like this. So if they're anchored in this membrane, let me just draw this as a double membrane here. Right? If this is anchored here, it has this transmembrane domain. Usually this is in the form of something called immunoglobulin class D, but there are 5 different classes of these immunoglobulins. Okay, this is called an immunoglobulin. Now, when an immunoglobulin, or we call this an IG, which I'll write down in our abbreviations in just a second, when this is anchored on the surface of B cells, let's actually say on the surface of naive B cells, we call this a B cell receptor. B cell receptor. And I'll tell you, even though we're going to use that term here, in immunology is very rarely used. And I'm just going to indicate also, even though you don't understand this quite yet, most commonly of class B. But when that B cell has encountered antigen, has been activated and undergone clonal expansion and has become an effector cell, then that immunoglobulin becomes secreted. So it loses its transmembrane domain and it becomes a secreted immunoglobulin. So when this is secreted from, and I'm gonna, whoops, form. From an effector. These guys are called plasma cells when they're secreting antibody. Then they're called, these particular molecules are called antibodies. Antibody, or AB. Okay. These immunoglobulins, again, they have some very particular features to them. First of all, there are two heavy chains and two light chains. And these guys are bound together by disulfide bonds, SS here, and SS here, disulfide bonds. So there's one link here, and one link here. This region here is called the hinge region because it's exactly that. This is where that molecule can kind of pivot and gain some flexibility here. There are some other really key regions on this molecule where the heavy and light chain come together here. This is the antigen binding region or antigen binding domain. And this whole top portion here, therefore, is called the FAB region. This stands for functions or functional antigen binding. Right, FAB region is this region at the top. And this is what varies from one type of molecule to another. The next part here is, oh, dinner. The next part here is called, yeah, I'll get on that. The bottom portion here is called the constant region, or FC. Right, functional constant region. The constant region is really important in binding to the surface of white blood cells. So the constant region is where we have recognition by white blood cells. And usually in the form of something called FC receptors. Note also, of course, the FC portion is also what has that transmembrane domain, or there is a transmembrane domain that's bound to the FC region for these anchored B cell receptors. The constant regions, even though it's constant, so you would imagine it's constant, it actually has 5 different classes, right? These constant regions. So there can be 5 different types of molecules of these immunoglobulins. So 5 genes that make up that FC region, only one of them is made per B cell, and that B cell can switch from one class of antibody to another class of antibody, or one class of immunoglobulin to another class, and actually receives input from whatever's happening in the surrounding and where that activation of that B cell occurred. For example, for my studies, I study immunoglobulin class A because I study mucosal immunology. So stuff that's in the mucosa, in fact, coronavirus, for example, is a mucosal pathogen. And it seems that the best antibodies to respond to that pathogen are immunoglobulin class A, because these are the only ones that can get out to that external region in the mucosa, while IgA and immunoglobulin class M are the two mucosal antibodies, right? So the 5 classes, this is where we have the ones you've heard of before. The classes would be class G, class M, class A, class D that I've mentioned already, and class E. And just super briefly, IgG is primarily responsible for systemic responses. That means something in circulation, in your blood, in your tissues, something like that. IgM is a mucosal pathogen, or I'm sorry, is a mucosal immunoglobulin, but also is the first antibody secreted anywhere in systemic response or mucosal response. So well, that's not entirely true. For systemic response, it's the first antibody secreted. IgA, this is mucosal. IgD almost always is a B cell receptor, and IgE is involved in allergies. Now we'll talk about these more in the second sets of slides, but just so you have a general understanding, there are 5 different genes that make up that FC portion. Only 1 gene is turned into that protein per B cell at any given time. And so that B cell can be secreting out IgG, IgM, IgA, IgD, or IgE, but most of the time it's always confined to whatever the response is that is needed or wherever that B cell has been activated. Okay, so that's FC and FAB. Now the development of the FAB region is what we'll talk about next. How these immunoglobulins gain their massive variation at this FAB region. Well, I think that's the majority of the stuff. Let me go back to our slides and just see what the slides have to say about these B cell receptors. Okay. Again, immunoglobulin is what this thing is called. When it's on the surface of the cell, it's a B cell receptor. When it's secreted, it's called an antibody. And this is from, I don't like the word mature, it should say an effector plasma cell. This is a glycoprotein. All of them are. The immunoglobulin that I study, IgA, is very, very glycosylated. There's a whole bunch of different regions that have glycans on them. Oh, all of them have glycans on them though, and I'll tell you that there's a whole field of study on the relevance of these glycans on the function of these proteins, but we won't talk about any of that stuff here. I've just mentioned all of these different regions, the antigen binding site at the tip of the heavy and light chains on either side, and the differences between the variable regions and the constant regions, or the FAB fragment versus the FC fragment. This is how this looks as an anchored protein. Now I will also mention that B cells have MHC classes 1 and class 2, which will become important when we talk about how B cells become activated in the secondary lymphoid organs. But for now, we're just focused on those antibodies and immunoglobulins. Okay. Again, there's the 5 classes, IgM, IgG, IgA, IgD, and IgE, and the classes are all determined by the FC portion of the heavy chain there. The disulfide bonds that are formed here and here and here, and there's 2 of them here at the hinge region, but this hinge region provides that flexibility to that immunoglobulin molecule. All right, that antigen binding site has to have that perfect match with the antigen. But that constant region is the same for all of the antibodies within that class. So let me switch back over one more time to our little abbreviations charts. And we'll just add a few things in here. So Ig, immunoglobulin. This would be either a B cell receptor or secreted as an antibody. And we have our FAB region, which is the antigen binding region of an immunoglobulin, and the FC region, which is the constant region, binds white blood cells. of immunoglobulin. Okay. I think we're good. Okay, I'm going to pause the video here. Actually, I think I'll stop the video and I'll pick back up again on diversity and specificity, and then finish up this first half of the video with the antigen presentation. But I think that this video I'll have to pause here. I got to go make dinner for the family. So I'll come back in a bit with the second round of this video. Okay, let's finish up these first sets of slides on Chapter 13, the adaptive immune system, and we're going to go back over to our slides and look at the development of the diversity of the receptors for both B cell receptors and T cell receptors. Now, I've mentioned to you already that our adaptive immune system has the ability to essentially recognize and respond to any pathogen that's on our planet, whether we've seen it or not, right? And if you really think about that, it is a mind-blowing concept that our B and T cells, without ever seeing anything come into our body, has the capacity or the potential to respond to anything that comes in. And you know this is successful because we're still alive, right? As a population, as a species, but even as individuals, we come in contact with pathogens constantly, and we're able to mount an efficient immune response, not because our parents did, but actually because our cells have the ability to differentiate their receptors and make them so varied that they have the capacity to respond to anything foreign that comes in. So this is a discussion of how those receptors gain their variation, what's called diversity, and gives us, again, this term repertoire. So that whole collection of diverse types of things in our closet. So this is a two-part selection scheme. The first is without the presence of any antigen. This whole concept is called the clonal selection theory. Clonal selection theory. And again, there's two arms to this theory. The first portion here involves just a genetic selection process and then essentially boot camp training inside the primary lymphoid organ, selecting for only those organisms that are not self-reactive and also able to understand what is self from non-self. So in the, and this, I'm sorry, so that's part one here, this top aim. And then once you have the development of this diverse receptor repertoire, and these guys go out into the secondary lymphoid organs, and then they are selected upon by the presentation of antigen. So only those cells that have the receptors able to bind to with perfect specificity to particular antigens that your body is facing will then be selected for and allowed to amass a giant army of clones. So this early part, this early arm is happens in the primary lymphoid organ, where that lymphocytic stem cell, as you remember, that one that was derived from the pluripotent stem cell, divides into B and T cells, and then those then further differentiate. And the differentiation within the T cell arm and within the B cell arm is the varied types of receptors of that particular variable region on either the T cell receptor or the FAB region, that antigen binding cleft on the B cell receptor. So during that development and differentiation, so as that stem cell differentiates into the various types of B cells or T cells there could be, there is genetic changes, there are genetic alterations that happen upon each new generation that's made, that essentially is rearranging all the genes that are responsible for putting forth those receptors, those immunoglobulins for B cells or T cell receptors. And that rearrangement produces an immense variability. In your textbook, unfortunately, they've minimized this to like a little tiny box in the window. So I'm just going to expand on that just a little bit. And these three concepts you should be able to understand. We're just looking at immunoglobulins here, but a very similar process occurs for T cell variable regions too. So this is how antigen diversity is developed in immunoglobulins. First, let's just look at the genes involved in the development of just that FAB portion, that variable region here in the immunoglobulin molecule. This is just looking at the heavy chain alone, but the same is true for the light chain. The variable region is composed of a variable sequence, the variable region gene. I'm sorry, the FAB portion is composed of the variable region gene, which is where that antigen binding site is. This D or diversity region gene segment or protein portion here a joining chain that joins this variable region with the constant region, and then the constant region transcript here that turns into a protein. So this is showing you the process of rearrangement, where if you just look up here, this is our genome. All humans have the same genome. We all have in our genome about 65 different genes that encode for just that top portion of the protein on the heavy chain. So over 65 different variable region genes. And for every B cell that's made, that B cell essentially will, I know these words are whatever, turning cells into humans, but we'll pick one variable region gene as the starting part of that, that receptor, that protein. One D region gene, so there's about 27 different diversity region gene segments. So one of those diversity region gene segments will be chosen. There are 6 joining sequences, and then there are, of course, those 5 constant region genes. Mu for M, gamma for G, I'm sorry, delta for D, gamma for G, epsilon for E, and alpha for, focus on these ones up here. So for every single B cell, there will be a different variable region combined with a different D region combined with a different J region. And so that if you just look at the statistical combination of the 65 variable region genes with the 27 diversity region genes and the 6 joining sequences, that in and of itself provides an immense amount, like 10 to the 9th possible combinations of just the different types of gene segments that come together to form just that variable region. So that's this first part here, the rearrangement of the genes for the V, D, and J regions. Together, this encodes just that heavy chain variable region. Now, the same thing happens on a light chain. So down at the bottom, combinatorial associations means combining one unique heavy chain with one unique light chain. And that combination in and of itself multiplies the diversity quite a bit. This middle part here has to do with reading frames. If you recall from chapter 8 on genetics, we talked about how if you start reading a sentence or if you start reading an mRNA transcript at reading frame 1 versus reading frame 2 versus reading frame 3, and the reason that frame shift mutations are so impactful on that final polypeptide is because the entire reading sequence will be completely switched if you read it a little from a different nucleotide position. So our cells actually take advantage of this to increase variability for that gene encoding segment. So by imprecisely joining that joining region where the variable region is joined to that constant region, or where each of these gene segments are joined together, with imprecise joining where a nucleotide is taken out or 2 nucleotides are taken out or something like that, then it shifts that reading frame so that that polypeptide sequence is slightly different. And this is really important in increasing that variability of what that binding site looks like, right? Because remember that structure equals function. So if you're switching the arrangement of the amino acids, and it's just for this ability to bind to and recognize any foreign peptide, then increased variation, so increased weirdness to that variable region is beneficial because you don't know what you're going to be faced with in the first place. So with the combination of all of these 3 factors there, rearranging the V, D, and J gene segments to provide you that initial variation, changing the way that these gene segments combine together by shifting that reading, or allows the shifting of the reading frame to create a totally different polypeptide product, even from the same V, D, and J gene segments. If you combine them together by missing 1 or 2 nucleotides between them, you'll completely produce a different protein product or receptor region. And then combining those light chains with the heavy chains in different combinations makes it so that the possible combination of these different clones would be 10 to the 14th to 10 to the 18th. And to figure out those numbers, 10 to the 18th, that's a 1 with 18 zeros behind it. 18 zeros. A million is a 1 with 6 zeros behind it, right? A trillion is 1 with 9 zeros behind it. So to get 18 zeros behind that 1 is such a mind-bogglingly giant number. And all of these is just without the presence of antigens. So all of these possible clones are generated that are able to be reactive to whatever. Now, there might be a problem here though, because with all of these possibilities, certainly there will be some receptors that have self-antigen recognition. So the problem here, of course, is the ability to react to self. Now, this is a substantial problem, and you probably know what this is called. There's a baby crying back there. When there is self-reactivity, this would be the development of autoimmunity or an autoimmune response. So in order to prevent that from the get-go, there is a negative selection process that occurs for both B cells and T cells, and this is still during the development of these initial cells before they get out into circulation. So the solution for self-reactivity is a process called clonal deletion. Now, this term clone, just think about this as, remember that a B cell, from one neighbor to another, they're essentially clones of each other. They just have variable regions. They just have different types of immunoglobulin variable regions. So we can use the term clone to describe them. Usually when we're talking about clones, we're talking about when you take one B cell and you actually propagate it to make exact copies of itself. That would be a clonal population. So when you hear this term clone, just kind of keep in mind, we're kind of just talking about the B cell or T cell repertoire, or we could be talking about propagation of an individual cell. Anyway, this concept, clonal deletion, or also termed negative selection, is when cells that are self-reactive are deleted. I think I have a picture of it back over here. Let me just show you. So in this picture here, this step number one essentially is that genetic rearrangement of those gene segments, all the stuff that we just mentioned, to give the diversity in these B cell receptors or immunoglobulin receptors. And then the second process here would be clonal deletion, where these really specialized cells come along and present self-antigen to all of our clones that are made, and anything that is self-reactive is immediately deleted. So that by the time the army is ready to leave that primary lymphoid organ, we know that we are only releasing lymphocytes that do not react to self, ideally. Now, this is not perfect, but it is ideal that that's how it happens. For T cells, they have to also undergo positive selection because remember they have those CD4 and CD8 co-receptors. And it's super important for those co-receptors to bind to and recognize their particular class, MHC class receptors, so that you don't have or accidental activation of, say, a cytotoxic T cell by an antigen presenting cell. T cells have that extra step of positive selection. And I'll say that the checks and balances in our immune system from development to activation actually are quite extensive. And there are a whole bunch of different roadblocks along the way, especially for B cells, that they have to pass through and before they can actually get out. And then once they're out in circulation or at the secondary lymphoid organ, even there they have a couple more checks and balances. So constantly along the way, these T cells or B cells are just deleted left and right so that they reduce the chance of autoreactivity or self-reactivity. In the thymus, they have these amazing cells. I forgot the name of them, but these amazing cells, the only kind that exist, there's something similar in bone marrow, but in the thymus, they have these cells that are able to essentially produce proteins on the entire genome, which if you think about it is really unique because every single cell has massive gene regulation on it. And which is why our skin cells, our keratinocytes look totally different from our dendritic cells, look totally different from our neurons, etc., right? Because of gene expression. All of our cells have the same genome, but because of regulation on their, oh my gosh, I bet you haven't been hearing any of that stuff. Shoot, I might have to go and redo this whole video. Because of regulation, I forgot that I had you guys on my earbuds. Because of regulation, all of our cells look different, but in the thymus, because of this concept of clonal deletion, that really we need to make sure we have no receptors out there that have the ability to recognize any self proteins, that these one unique kind of training cells essentially just go through the whole genome and continuously produce proteins that are just on the human genome, just to make sure that we have no self-reactivity. Now, if you can imagine, I just wanted to kind of segue a little bit. There is some problem with this because there are some proteins that are not produced by those initial training cells that really you would never see unless maybe a cell lysed open or was not apoptotic, but was necrotic, so had a tissue damage like cytotoxicity and came apart. Which does happen on occasion, but not usually. Usually in the body, we have very controlled cell death, which eliminates inflammation. to allow for regeneration. But on occasion, if there's cytotoxicity or some kind of damage, then you might have, the body might see, or the immune system might see proteins that it never would otherwise, because usually they're in these kind of hidden locations within these cells. So there's a slight problem to that. It's not a perfect process, and there can still be some cells that get out that are autoreactive. And even downstream, the development of an immune response to viruses or bacteria that have antigens that look really similar to ours can induce that autoreactivity too. But we do have a pretty good process in place to this clonal deletion and positive selection before things leave that primary lymphoid organ. So these last terms here, I think we're close to the end, but these terms are really important to understand as we move into understanding the challenge and the activation of our B and T cells. When these guys are in this development process in the primary lymphoid organ, before they have developed antigen receptors, we call them immature lymphocytes. These vocabulary terms are important for you to get, so please get these ones down. Immature means they're underdeveloped antigen receptors. Once they've undergone development of the receptors and they've been selected, so both positive and negative selection, we now call them naive lymphocytes. And these guys are the ones that actually leave the primary lymphoid organs and arrive in the secondary lymphoid organs. They have antigen receptors, but they have not yet encountered antigen, right? So they're called naive because they haven't seen their cognate antigen yet. Once they're in the secondary lymphoid organ, if they come in contact with antigen and they bind it, they are activated, and these are ready for proliferation. So this is this really brief intermediate step. When they find their antigen that they recognize, they bind to it, their match is made in heaven, they have check 1, check 2 in place, and they are ready to divide and conquer. These are called activated lymphocytes and still within that secondary lymphoid organ. Now, once they proliferate, once they start replicating and making giant clones of themselves, there is a differentiation that happens from that first cell to all of its progeny to make effector cells and memory cells. The effector cells are ones that are descendants of the activated ones. Excuse me, we call them terminally differentiated. So they have reached their climax, their apex. That's what they're going to be. And now they're going to go make an effect. They're going to go do whatever their job is to go do. Some of them stay inside the secondary lymphoid organ, depending upon the nature of that particular cell. Many will leave the secondary lymphoid organ and migrate out to the infected tissue and do their effect there. The memory lymphocytes are also progeny of those activated lymphocytes. They look exactly like that initial activated lymphocyte, but they're partially differentiated. So they kind of stay in this memory state. They're long lasting. Sometimes they can live for the entire rest of that person's life. Sometimes it's only for a short window, but they stay in this partially differentiated state. And those are what provides that memory or amnestic response the second time around. They can become activated instantaneously. So these terms are important, and they'll help us get through our understanding of the activation, challenge, and activation of these B and T cells. But the last concept in these slides is the presentation of antigen. I think I'm only going to just mention this real briefly, and then we'll end this video. And hold on, I got running kids here. Just a minute.
I won.
Go. Guys, I'm recording.
Oh, hi, everybody. My name is Ryan Dunn. That's Jada Hamilton. And we're this person's daughter and son.
Yeah, we have different last names.
Yeah.
Make sure you include that.
Go on in. I'm recording.
And Ryan is not Jada's brother.
Close the door, honey. I think I'll have to re-record that part. Okay, so we're talking about the presentation of antigen, and this would be by antigen presenting cells, as we've talked about many times. We are primarily going to focus on dendritic cells because these are the ones that activate the naive response, the naive T and B cells. But there are other antigen presenting cells and macrophages we've mentioned already too, and even B cells. And this will become really important when we talk about B cell activation, because they need to be antigen presenting cells in order to become activated. But macrophages are responsible for activating the memory T cell response, which again is a very important response. These guys, of course, we know are phagocytic cells, so they engulf and digest as phagocytes. They process those antigens and then they bind antigens to that MHC class 2 molecule and present it on the surface. And actually, this is where I'm going to kind of give you a little hair-brainer thing. So even though this is from your book, this shows you this whole concept of a dendritic cell phagocytosing some foreign molecule, breaking it apart, taking those antigens and presenting them onto MHC class 2. And during this process, they're migrating to the secondary lymphoid organs where they can come in contact with T cell and form that match made in heaven that we talked about before, that MHC class 2 presenting foreign antigen that binds to a T cell receptor, remember, with that variable region, and that CD4 co-receptor recognizing that MHC class 2 complex. And this will lead to activation and then clonal expansion of this T cell. Now, I will just mention here, and then I'll just have to share this on the next time around, but there is something called cross-presentation. So the same concept must occur to CD8 positive T cells as well. So we'll discuss what cross-presentation is and what that looks like in the next sets of slides, and probably we'll re-record this whole thing. So that's it for this one, and we'll pick back up again right here on the next video.